Also, before I forget, just to give you guys a heads up, if you're looking for volunteer service type hours, now that we're back in session here, Saturdays at 11 is the MS Aquatics program, so that's still going every Saturday. And I just got an email from the instructor, Annie Foose is the instructor, says, hey, we'd love to have more people out or anybody that's interested. So it's the Recplex. You can just show up. Some of you I know have already participated, right? So yeah, that'd be great if you're interested in doing that. You can just give me your forms and I'll sign them whenever after you've done a few sessions, whatever, whenever you feel like you need to, I'll sign those forms for you. I can also send out, I don't know if Jennifer Westover is a third-year student who's kind of the coordinator for that program, the student coordinator. She's there on most of the weekends. But she has a calendar, that Google Calendar. I don't know if she shared that with you in the past. But I can either send another email out with a link to it again, just to remind you. But and then you can at least take a look and see if other people have signed up, so you can spread yourselves out to help you kind of do that a little more evenly. So anyway, I appreciate that. Thanks. So synapses and synaptic transmission. So you learned on your own about action potentials and so forth, and generating those signals that are going to then typically lead to some release of neurotransmitters at one of these synaptic clefts or junctions, and it's going to help communication with either another neuron, a muscle, a gland, or something like that. So that's what we're talking about today, is how that signal transmission happens here between actual cells. So we're talking about the synapse, that specialized point of contact between a neuron, another neuron, or a muscle cell or a gland. Okay, so our neurons do communicate with other than just neurons. Information is transmitted from one neuron, called the presynaptic neuron, to the postsynaptic cell or neuron or muscle or gland. Okay. A couple types of synapses that we'll talk about. One, very briefly, is the electrical synapse. Okay, so we do have electrical synapse, which are also referred to as gap junctions, are what are used to transmit this electrical signals directly through between the cell membranes. You have a pre and a postsynaptic cell membrane that are really close together. They get really close together, and you have these pores or channels made up of proteins called connexins. Okay, it's a channel protein that allows the direct flow of ions and current from one cell to another. Okay, so they just right through an open tube, basically. Okay, big and fast. So these are important, especially when cells have to coordinate together and contract at the very same moment in a very coordinated fashion. So when you think about like the heart muscle, so the heart muscle this is how signals, once the heart muscle is stimulated between the actual heart cells, muscle cells, it's through these gap junctions. So you get this wave of depolarization that happens quickly and creates that contraction in your heart in an appropriate way. Okay. And some other smooth muscles as well do the same thing, and even some glands. And I added this last one is glial cells. Remember we said glial cells, the glue of the nervous system, okay? The neuroglial cells, they also have some of these gap junctions. And so some molecules can move between the glial cells, and you can even have waves of electrical impulses move through glial cells. They're not truly generating an action potential, but that's how some of this communication there's theorizing can occur in glial cells, is through these electrical synapses. Just showing you here a picture of that really narrowed point between the two membranes, pre and postsynaptic membranes, and then these connexins that are forming these open pores that allow ions and other small molecules and even some electrical current to pass through those pores. Any questions on that? Electrical synapses. We're going to spend the rest of our time focusing on chemical synapses. This is how most of the information in the nervous system is transmitted from one neuron to another. It's through a chemical type synapse. You have a presynaptic terminal in one neuron. Some synonyms for presynaptic terminal is also called the axon terminal sometimes also referred to as the terminal bouton. Okay, so if you hear any of those, presynaptic terminal, axon terminal, terminal bouton, we're just talking about that presynaptic cell's endpoint. Okay, then you have some type of space, a cleft, a synaptic cleft, and then you have your postsynaptic cell membrane, and so it's the postsynaptic terminal, the membrane of that target neuron or target cell. Okay, so I shouldn't just say neuron, it could be a muscle cell, could be a gland, but it's that postsynaptic terminal area, membrane area on that cell. You got to have, it's a chemical, so you got to have some type of chemical. In this case, we're going to be talking about neurotransmitters. Okay, so neurotransmitters are chemicals contained in small vesicles that are then used for delivering those across that synaptic cleft for communication. We can have different types of connections. We can have what are called axosomatic types of synapses, axodendritic, and axoaxonic. Okay, so if you just look at the breakdown of the name, axosomatic means you're going from the axon of one neuron, and that terminal bouton or that presynaptic terminal is communicating directly with the cell body, the soma. An axodendritic is the axon or that presynaptic terminal is communicating or touching another dendrite of another neuron. And then axoaxonic, it's actually directly connecting the end or terminal end of another axon. Okay? So this is just showing you a very simplistic diagram of this arrangement of our presynaptic axon, our synaptic cleft or space, and then the postsynaptic receptor area with its receptor sites, the postsynaptic membrane, and our little vesicles with some neurotransmitter inside of there. And then this just is representing the different types of connections that can occur. So on the top here is the axosomatic. So you see there's the connection here between the axon of one neuron with the cell body of the other neuron. In this case, the axon of one neuron is connecting or communicating more with the dendrite. And then down here, you can see here's the axon, terminal axon of one neuron communicating with that terminal axon of another neuron. Okay, so that's axoaxonic synapses. So going down the events of what occurs during this process of synaptic transmission. First of all, you have an action potential that's been generated in a neuron somewhere up the stream. Okay, that action potential is causing depolarization and is causing voltage-gated sodium channels to open, causing depolarization to go down to the end of the axon terminal. Okay, when it gets to the very end of that axon terminal, that presynaptic terminal, then we also start to open voltage-gated calcium channels. Okay, just like there's voltage-gated sodium channels, we have a little concentration of voltage-gated calcium channels at the end in that terminal, that presynaptic terminal. So that causes the calcium channels to open. Calcium comes in, okay, and calcium triggers the little synaptic vesicles. I don't know exactly why or how, but the little synaptic vesicles that are sitting in that presynaptic terminal or terminal bouton, whatever you want to call it, to move to the membrane and open. In the process of exocytosis, release the neurotransmitter into the synaptic cleft. Okay? Those neurotransmitters go across the synaptic cleft and bind with the postsynaptic membrane receptor. There's hopefully receptors there that will want to bind that particular neurotransmitter. The postsynaptic membrane channels then change shape, okay, and allowing ions to enter the postsynaptic cell. That's one thing that can happen, just directly opening some ligand-gated ion channels, okay. So when something binds, so in the case of a neurotransmitter, when we talk about the types of receptors that it binds to, typically as a big class, you call them ligand gated. So anything that binds a neurotransmitter to it is called a ligand gated channel. So these neurotransmitters bind to these ligand gated channels. They open and they can either cause depolarization, depending on the neurotransmitter, or it can cause hyperpolarization and actually cause it to get more negative. So you can either cause excitation to the next cell, or you can inhibit the next cell, depending on the neurotransmitter that's being released. Okay? Or you can activate one of the G protein mediated receptors. If this is a neuromodulator that's being released, you might, instead of just opening up a ligand gated, quick acting, protein channel, you might activate what's called a G protein mediated receptor. That's going to cause more long-term changes to the cell, not just a quick opening of a protein channel that allows some very brief changes in polarity. You might actually be changing how the next cell functions through these more complex G protein mediated pathways. Okay, and that's usually through what we call a neuromodulator versus a more fast-acting neurotransmitter. Once it's bound to the neurotransmitter, it's only there temporarily. It's released, and it's either broken down in the synaptic cleft, okay, and reabsorbed and not repackaged, but some of it is repackaged, some of it's reabsorbed. So sometimes the presynaptic terminal will take back up some of the leftover neurotransmitter and then repackage it or send it back up to the cell body to be repackaged. Okay. And that's the form of endocytosis, that's this reuptake that occurs. But some of it's just broken down in the synaptic cleft, never to be seen of again. So, and that ends the transmission of what's happening once everything unbinds from the channel. So here we have our action potential coming down a neuron, down the axon to the axon terminal. You see our flip-flopping of charges here. You see the charges going back and forth here. So we're depolarizing here, and we're coming to the end of the axon terminal. We have our synaptic cleft, we have our postsynaptic membrane, and we have some receptor sites in yellow there, some protein channels that are ligand gated. Once we get this depolarization here, so the inside of the cell gets more positive here, depolarized at the presynaptic terminal, that opens up voltage-gated calcium channels. Okay, they respond to that change in charge, and that allows calcium, because there actually is a calcium pump too that keeps calcium concentrations higher on the outside of your neuron as well. That allows now calcium to come in. Calcium triggers these synaptic vesicles that are close, but not quite, touching the presynaptic membrane to move towards the edge and release their neurotransmitter. So here they are moving towards the release sites, lining up there, ready to go. And they release the neurotransmitter into the synaptic cleft. Those bind, find their way to their appropriate locations, hopefully, and this one may eventually bind up another one. And when that occurs, that opens this ligand-gated channel, so it opens, and that could be a sodium channel, and it might allow positive sodium ions to go in and get the next cell to start depolarizing in preparation potentially for an action potential. Or those might be chloride channels, and it might let in negatively charged chloride ions and make that next cell hyperpolarized, so it gets more negative and it's less likely to cause a contraction, so, or not a contraction, but an action potential. So we don't know enough. We don't know what neurotransmitter this is. We don't know what's going on here. This is an excitatory or an inhibitory connection here. Well, I guess we do now. So this one's showing this one's must be some positive sodium channels open, so they're letting positive charges in and enter into the cell. So this is actually causing what's called an excitatory postsynaptic potential. There's the potential to cause an action potential, but we've got some excitation going on, but we may not have reached that threshold value, of minus 50 millivolts or so. We may not have gotten to that threshold value to get all the voltage-gated sodium channels to start to open. Okay, this is some ligand-gated sodium channels, but we may not have got to the threshold stimulus. Okay. So let's talk about these local potentials that occur at these synapses, okay? As I mentioned, you can either get an excitatory postsynaptic potential. That's what this is. If someone happened to release some positively charged ions were going into the next cell, that would likely be excitatory, right? Because it's depolarizing the neuron, the neuron next in line. So that's an excitatory postsynaptic potential, or an EPSP. Here's the S. Excitatory postsynaptic potential. There we go. So that's called an EPSP. So you get some depolarization. If you get enough depolarization, typically through the process of summation, because maybe there's some other axons nearby that are also connecting and delivering neurotransmitter at a similar time, the summation of all those positive charges coming in gets you to the threshold stimulus, and then all the voltage-gated sodium channels open up. And so then that would lead to an action potential, okay? But in this case, we're just talking about an excitatory postsynaptic potential. On the flip side, we can also have neurons that inhibit neurons, okay? And that's via an inhibitory postsynaptic potential. In that case, you would typically allow some type of a negatively charged ion, most commonly chloride, to come in to make the inside of your next neuron more negative, so it's less likely to fire, okay? Another way you can get your neuron to become more negative is to open up a few potassium channels and let some more potassium, even though it already wants to leak out, right? It's got those leak channels. If you open up some more, it'll even leak out more, and you're losing positive charges out of the inside of the cell to get even a little more negative. So those are the two ways we can make the inside of the cell a little more negative. Either bring in some more negative charges or let out some more positive charges, okay? And that makes your next cell a little more inhibited, less likely to generate an action potential, okay? We always, with all of our systems in our body, including the nervous system, you have to be able to turn it up or down, right? You know, you excite it or you inhibit it. Usually most of our body processes are set somewhere in the middle. You know, we're not turned all the way up or turned all the way down. We're somewhere in the middle, and that's true of our nervous system. We wanted a certain level of activity for normal movement, normal activity, but we want to be able to ramp it up for certain things or turn it down for other things. So we have to have both excitatory methods or impulses and inhibitory ways to influence the nervous system, okay? And these two processes together regulate the excitability of the nervous system at any given moment and any given location within the nervous system, okay? So this is just showing you on your left there, this would be an excitatory, EPSP. Here we have the cell membrane. Here we have the negative. It's negative on the inside. You have a neurotransmitter that binds, that opens up a ligand-gated sodium channel. A few sodium ions move in. A few more positive charges are coming in here. So here you see the resting state is minus 70 millivolts. You let a few sodium ions in. It goes up a little bit, maybe to minus 60 millivolts. It's a little more positive or a little less negative on the inside, but it doesn't generate an action potential because you didn't reach that minus 55 millivolts of the threshold stimulus to get the voltage-gated sodium channels to open. Does that make sense? So you just opened up a few ligand-gated here, and then it went back down once those sodium were pumped back out by the sodium-potassium pump, okay? Here's the opposite of this is an inhibitory postsynaptic potential, an IPSP. In this case, this neurotransmitter happens to fall on some chloride channels, and it opens up and allows some negative chloride ions to come in and make it a little more negative on the inside. Yeah?
So both the excitatory and inhibitory postsynaptic potential, they can both use temporal or spatial summation to achieve whatever.
Yes. Yes.
So the EPSP and IPSP, they're both potential, but they're not the same as spatial and temporal summation. But it looks like that's what they can use to achieve what they're trying to do. I just don't really know the difference between the local potentials and spatial.
Okay, so local potentials are the little changes in the resting membrane potential at that local region. Local potentials, when added up either temporally or with temporal summation or spatial summation, usually a combination of the both, when you add them up, they can then lead to an action potential if they exceed threshold. If you get enough small local potentials that occur on the same neuron, close to each other at about the same time, then you might achieve enough of a decrease in enough of a depolarization of that cell to reach that minus 55 millivolts. Does that make sense? You also have an excitatory postsynaptic potential. You get enough of those excitatory postsynaptic potentials occurring at the same location at about the same time, so both temporal and spatial summation can lead to an action potential. Inhibitory postsynaptic potentials can also be summated, and it just means that that's going to get even more negative and less likely to have an action potential or get excitable. Okay, so they can all add up as long as they're occurring at about the same location at about the same time. Does that make sense? Okay. All right, so that's just a little overview of the basics of little excitatory postsynaptic and inhibitory postsynaptic potentials. And so now let's talk about this concept of presynaptic facilitation and presynaptic inhibition. This is a concept that when an amount of a neurotransmitter that is released is either increased or decreased by the influence of a previous activity of an axo-axonic synapse. So all that we're going to talk about now is just different ways that the nervous system shapes output, and just a little more nuanced in how the nervous system can influence other neurons within the system. Okay. Most of this has to relate to things like neuroplasticity and learning of new material and how our brain and nervous system shape itself in response to repeated stimuli. These types of influences we're going to talk about can either be strengthened or weakened through use or disuse. And these are things we have where we talk about motor learning or neuroplasticity in response to recovery of function after a stroke, or even things like chronic pain. Those kind of things can be influenced by these types of connections within the nervous system. They can either go haywire and mess you up, or they can be helpful for what you're trying to do. So, so the concept of presynaptic facilitation is where a presynaptic axon releases neurotransmitter that causes slight depolarization and typically some influx of some calcium, prolonging the action potential when it arrives at the terminal. So here we have, this is our presynaptic neuron. Our primary signal is coming down to communicate with this next neuron and cause release of neurotransmitter. Okay. Here is another neuron that's nearby that is communicating with this presynaptic neuron via an axo-axonic synapse right before that signal gets there. And what happened is this neuron has sort of primed the pump, so to speak, already. So this neuron, this presynaptic neuron here, has released a neurotransmitter that happened to cause some calcium to already start to get into that presynaptic terminal, so that when this action potential arrives, even though an action potential is an all or nothing response, so that action potential gets there, there's already some calcium in there. It's already a little more positive. So that when it gets there, even more calcium is going to come in, and you're actually going to probably get even a stronger or larger release of neurotransmitter at that synaptic cleft than if you didn't have this guy helping it out. Okay? So basically, this just is giving this neuron a little bit of a helping hand to amplify the signal when it does get there. So you tend to release a little more neurotransmitter than if you just sent that action potential alone and didn't already prime it with a little bit of positive charges before it got there. Does that make sense? And so that is one way that all these, when we talk about interneurons, most of our nervous system is interneurons. So these guys here, the ones that these doing this presynaptic facilitation, and they can also do presynaptic facilitation, that's often done by a lot of the interneurons in the area. The interneurons will influence the connections and circuitry going from these longer neurons that are communicating over longer distances. But regionally, you have all these interneurons in the spinal cord, in the brainstem, that can cause and have these influences over the excitability of that circuitry. Okay? So it's just a way of priming the system or making that neuron a little more active based on some neurons around it that are communicating just before that synaptic cleft. Okay? That's called presynaptic facilitation. Presynaptic inhibition is the opposite. Okay? So in this case, same kind of setup here. Here's an axoaxonic synapse. It has a neurotransmitter that binds here, but instead of causing calcium to come in and depolarize, it may actually cause something like chloride to come in and make that slightly more negative down here at the terminal bouton or the axon terminal. So when that action potential gets there, it just doesn't create as strong of a response. And you get less opening of calcium channels when that action potential gets there because it's a little more negative down at that axon terminal. And so you just don't get as many neurotransmitters being released. So you see only 3 there compared to the one before where you got more than double that. So that's just called presynaptic inhibition. I mean, of course, we could still have all kinds of other things coming in here to this cell from different locations that could either have positive or negative characteristics or influences on it. It's all a matter what the sum net result is of all the influences in a network of neurons at the same similar time, okay, when signals are arriving or moving through that area. And so all we're just talking about is ways of turning up or turning down the nervous system, and one of those ways is through presynaptic inhibition or facilitation. Not everything's just through its direct connection. Okay with that? Now we're going to talk about more the receptors themselves, okay, the synaptic receptors. What we focused on to this point is primarily what we call the ligand-gated ion channels, okay? These are the proteins that function just like a gate, okay? And the gate opens when you bind the neurotransmitter, and then when the neurotransmitter unbinds, the gate closes, okay? So that's how that works, ligand-gated. Pretty simple. Fairly fast acting. That neurotransmitter doesn't stay there very long, okay? The majority of synaptic activity is mediated by ligand-gated channels. So the neurotransmitter is the ligand, okay? So that's what I mean there. Binding to a protein channel, causing a conformational change in that protein channel to allow it to open up. And then when it releases after a few milliseconds, then it closes again. The other type of synaptic receptor is the G protein mediated receptors. That's the other major classification of receptors, okay? When you're just trying to send a quick signal from your sensory receptor up to your brain to say, aha, I feel a sharp tack on the bottom of my foot, that's mediated through those fast ligand-gated, open up some sodium channels, get a depolarization, get an action potential, send the signal to where it's going. But when you start using neurons and you start having things like chronic pain, maybe that you never get rid of that needle in your foot, and you've been walking around it with a month, and you've been sending signals for a month on that, and now all of a sudden you're developing like these characteristic limp to get away from it, and now you've also got this chronic pain response, and you've got emotional problems because of it. That's via all the other neurons of network of neurons being influenced by these G protein mediated receptors, and you actually change neurons to be more sensitive than they normally are. You get them to build new dendrites and axons based on all this chronic pain that you may be having, or if you're learning a new skill or activity and you're trying to actually get some neuroplasticity occur, the communication and connections in the neurons that are happening over longer periods of time, and you're wanting to get more longer permanent changes in how networks of neurons communicate, that's via these G protein mediated receptors. Or you have things that are happening that last more like several minutes to several hours and so forth. Okay, so that's through the G protein mediated system. More complex, slower acting, generally longer acting modulators of neural activity. So they tend to influence bigger networks of neurons and overall modulate excitability of networks of neurons and systems versus just the individual connections between neurons. Okay? The G protein mediated receptors are also involved in what's called the second messenger system. And we'll explain that in a second here. Here's one example of how these G protein mediated receptors work. So here's a neurotransmitter binding to a receptor, to a G protein mediated receptor. Okay, so when it binds, it will typically activate a G protein. Depending on the schematic you look at, and in your book, they show this G protein actually being like attached to the receptor. It doesn't have to be attached to the receptor. It may just be nearby. I'll show you a picture in a second that shows a different way of looking at the G protein. G proteins are usually made up of several subunits. In this case, it's an alpha, beta, and gamma subunit here. Don't have to know the detail of that necessarily. Just know that there typically is some subunits to that. In this case, what this is showing is when the receptor binds, it causes a conformational change in this case, the alpha subunit. Don't need to know the details. That conformational change allows the alpha subunit to actually move in the cell and go over to a protein channel and actually activate it. And it actually makes that protein channel open. And in this case, it's making it more excitable because it's letting in probably some sodium ions. However, this alpha subunit may actually stay there for a few minutes. It may actually bind to that protein channel and open it for several minutes versus your traditional ligand-gated, if the neurotransmitter just bound here, it was a traditional ligand-gated neurotransmitter-based protein channel may only open for a few milliseconds. This protein, G protein mediated pathway actually causes longer acting openings of ion channels. In this is through the alpha subchain. This is called alpha subchain activation. This is just one method. We'll mention some other methods how these G protein mediated receptors influence cell activity. Okay, this is one way. After a few minutes, it then frees up, goes back, and then gets reset. Okay, to be able to do that again in the future. Okay. So now let's talk about how the G protein mediated receptors involved in this thing called the second messenger system. This is where G protein activates an intracellular target protein. The target protein then produces what's called the second messenger. Okay, so the second messenger then can do lots of different things. The second messenger can open a membrane ion channel, just like this did. This is the alpha chain doing that. In this case, the alpha chain is kind of like the first messenger, but we'll talk about here what we mean by the second messenger in a second here. It can open a membrane ion channel. It'll typically open it for a little longer period of time than your traditional neurotransmitter. It may actually activate the second messenger that's produced, and actually that second messenger will go into the nucleus of the cell and actually activate genes that tell the cell, hey, you are now needing to do this, and I need you to build more dendrites, so I need you to go to the cell nucleus and tell it to start building more cell membrane and extend out over here to grow the axon or the dendrites over here, because now we're using this cell more often because we're learning this new skill or activity. This cell needs to be strengthened and built up, or you're now using it more often. We need to generate more neurotransmitter, so I need you to ramp up the, fire up the engines for generating more neurotransmitter. And so that can activate genes to actually do that, to make the cell change the way the cell functions based on changing its genetic activation. Or one of the main ways you can also just influence overall metabolism of a cell is by modulating the release of internal stores of calcium. We have internal stores of calcium in our cells, and so G protein mediated second messenger systems, that second messenger can tell your cell to release internal stores of calcium, and when you release those internal stores, it makes your metabolism of your cell, it's like firing up the engine of the cell. If you store more, then you decrease the metabolism of the cell. You release more, you make the cell more active. So that's the other way that you can influence cell activity via these G protein mediated second messenger systems. So here's a little more of a simplified schematic here. Here's our receptor in the cell membrane. Here's a neurotransmitter. And I would probably say, instead of neurotransmitter, we might want to say modulator because it's usually the neuromodulators, not the neurotransmitters, that activate G protein mediated pathways. You'll notice here's the G protein. It's embedded in the membrane. It's close by. It's not exactly touching the receptor, but that doesn't matter. This can still then cause that G protein to activate, whatever don't worry about the details of what that means, but you can see it has 3 different subchains here. One of those subchains then can go and activate an enzyme. And here, this enzyme is embedded into the cell membrane nearby. That enzyme then will cause the production of another cell product called the second messenger. Okay? So the first messenger is sort of the G protein. The second messenger is then the G protein activates typically an enzyme to then create, through its enzymatic activity, or build another product called the second messenger. Then that second messenger then goes to the cell nucleus and tells the genes to activate to do this, or it causes internal calcium stores to be released and causes the metabolism of the cell to go up. So it's that second messenger that actually does the actual final activation of whatever. Or that second messenger goes, maybe there's a protein channel here that's got a gate on it. That second messenger binds to the protein channel, opens it up. Okay, so that's the second messenger system. The G protein is kind of the first messenger, then the second messenger is the enzyme that's the product of whatever that G protein enzyme that was activated. Okay, so in your book, they have a kind of breakdown like this. So we have our traditional neurotransmitter, fast-acting, binds to a ligand-gated sodium channel or a chloride channel, whatever kind of channel you want. It opens it up. It allows positive or negative ions to flow in, okay? And that can cause local depolarization or hyperpolarization. Fast-acting neurotransmitter. That's the one we typically think of when we talk about nerves talking to each other, okay? Then we've got this whole other thing called G protein-mediated receptor pathways. That's these other ones. And they have these main different ways of influencing the neurons, okay, or the cells, the next cell. First is by the alpha chain or the second messenger. And either the alpha chain or the second messenger, if I go back here, this is the alpha chain activation. This is just where that alpha chain went over and opened up an ion channel. Or the second messenger from the enzyme producing the second messenger can go and activate the protein channel to open or close, okay? So that causes more prolonged opening of ion channels or closing. It can cause prolonged opening or closing, and can cause depolarization or hyperpolarization. So it can cause excitation or inhibition, but for a longer period of time, okay, directly by acting on one of the ion channels. The G protein mediated receptors can also, via just the second messenger system, activate genes within the cell itself to then increase rate of synthesis of specific cellular products so that the cell does what you think you need it to for the future, okay? Does it need to grow extra dendrites or axons, okay, because it's being used more? Or the second messenger can go and activate and modulate calcium levels inside the cell and just affect the overall metabolism of the cell, okay, and ramp it up or turn it down. If you activate a gene that tells you to build more stuff in your cell, you're going to also typically, that second messenger is also going to release more calcium stores in your cell to fire it up to start building stuff, okay? Vice versa, you might have a second messenger that goes and inhibits a gene and turns down calcium stores or restricts it so that you then turn down the cell and turn it so it's less active, okay? So on the right is the short-term just communication between neurons sending motor signals, sensory signals, okay? Fast-acting, short-term kind of stuff. All this stuff on the left is what happens when neurons and networks of neurons are working more on a long-term basis, especially during things like modulating long-term pain responses, motor learning, shaping bigger motor output over longer periods of time, okay? Does that make sense? Even just like learning processes of memorizing information. This is stuff that's happening in the hippocampus when you're trying to store memory. This is part of these G protein mediated types of receptors, and the binding of those are helping you to create the new connections in your neurons that say this is where your memory for this particular information you just learned today is, okay? So that's what's happening with these G protein mediated pathways. This is just here's a couple examples. Don't have to have these memorized. These are just some examples of second messenger systems, just to kind of make it a little more real. There's a second messenger system called the cyclic AMP second messenger system. So cyclic adenosine monophosphate, or cyclic AMP second messenger system. So in this case, what happens? This would be for your autonomic nervous system when you're wanting your heart to go faster. Okay, so you have your vagus nerve. I should say vagus nerve. It's actually not that's your parasympathetic system, but your sympathetic nerves coming out of your thoracolumbar area go to your heart. They release norepinephrine at your heart, right? So one of your sympathetic nerves goes to your heart. Norepinephrine will bind to a receptor that actually is a G protein mediated receptor, okay? The G protein then, what it does is it tells an enzyme in your heart muscle or heart cell called adenocyclase to make cyclic AMP. That's the second messenger, okay? Cyclic AMP then causes opening of calcium channels, which causes your heart to beat faster and causes your heart to contract more strongly. So that's using a second messenger system, okay? Because when you turn your heart on, it ramps up. There's a little delay ramping it up, and then there's a little delay turning it off, and that's because it's a little slower G protein modulated system. Okay, so it's not like it's instant and then instantly turns off. It's because it's using G protein mediated pathways. It's a little slower to ramp up and then a little slower to turn off. It's a little longer acting using the cyclic AMP system. Here's another example of a second messenger system that's used is the arachidonic acid second messenger system. In this case, for example, it's involved in an inflammatory process. So let's say you damage some tissue in your body, get an irritation of tissue, you get some damage going on. Usually you tear open some cells, so you whack your thumb, whatever, you get some tendinitis or something, you tear open some cells. Your cells, a lot of your cells have histamine in them. They store a little bit of histamine in them. So you release histamine into the surrounding area. Histamine then will fall on some G protein mediated receptors. Okay? Histamine then, once it falls on the G protein mediated receptor, the G protein then tells phospholipase A2, which is an enzyme inside the cell, to ramp up production of arachidonic acid. That's the second messenger. Then the arachidonic acid actually causes your cell then to increase production of a substance called prostaglandins. Prostaglandins then are released by your cell in that area, and prostaglandins bind to some of the receptors in your blood vessels and cause vasodilation and cause swelling and vasodilation in that area. And that's part of the inflammatory process, but it's a G protein mediated pathway. Okay? And so things like aspirin and other NSAIDs, they inhibit the enzyme, the phospholipase A2 enzyme, in this G protein mediated cascade. That's why you have less inflammation, less swelling, and so forth in that area with aspirin and other NSAIDs. Okay, and also histamine blockers. So, but that's how that affects that G protein. That's just an example. And there's hundreds and thousands of these examples. I'm just pointing out a couple that are more clinically relevant for you guys. All right, hopefully I didn't make something that should be fairly simple too complex. So let me know if that, if you have questions or struggles, and if you read through that information again, feel free to reach out to me. All right. Neurotransmitters and the receptors. So now we've talked about the receptors themselves, ligand-gated, G protein mediated receptors. Now we're focusing on the neurotransmitters themselves, and then the specific receptors to those specific neurotransmitters. So most neurotransmitters / modulators, okay, so we're going to kind of lump them together here for a moment, are classified as either an amino acid or a derivative, an amine or a peptide, okay. One exception is acetylcholine. It's a cholinergic neurotransmitter, acetylcholine. Neuromodulators, while technically under the broader classification of a neurotransmitter, tend to be longer acting and cause changes in larger networks of neurons and utilize these G protein mediated pathways. Okay? When you hear the term neurotransmitter, all these can fall under that classification, but we're going to tease it out a little bit more and go talk about more neurotransmitters versus neuromodulators. So these are most common neurotransmitters and neuromodulators. So the first is neurotransmitters. And I say fast acting most of the time. It's not 100%, but most of the time these are fast acting, bind to a receptor, cause a channel to open or close, and be done, okay? And quick acting. Acetylcholine is a neurotransmitter, but primarily in the peripheral nervous system, okay? As it relates to primarily at the neuromuscular junction, for contracting muscle cells, okay? You'll notice down here it's a neuromodulator in the CNS. It acts more as a neuromodulator in the CNS. These other three here work in primarily the CNS, and they are fast-acting neurotransmitters. And we're going to go through some of these individually in a moment, so I won't mention them right now. But GABA, glycine, and glutamate. So the nice part here, if you're trying to think, what are the neurotransmitters? Pretty much anything that starts with a G. And then you just, as long as you can remember acetylcholine also in the peripheral nervous system, but everything else is a G there is going to be your neurotransmitter. And then the nice part down here is nothing here has a G in it. So everything else is pretty much a neuromodulator. As long as you know that everything with a G is a neurotransmitter, everything else will fall under more of the classifications of a neuromodulator. And then you got acetylcholine here also that is in the CNS. Slower acting most of the time, G protein mediated. This picture here is showing you neuromodulators, the big ones. Serotonin, norepinephrine, acetylcholine, and dopamine. And the only thing I want you to appreciate here is that neuromodulators tend to affect larger networks of neurons. They tend to influence general levels of arousal, attention, emotion, even set states of the nervous system, overall tonal states of the nervous system, how turned on my muscles are at any one time to ready to go or not go, not the individual muscle firing, but the actual tonal sets, your vigilance, all those kind of things are kind of set by this overall regulation of the neuromodulators just influencing lots of neurons, either ramping them up or turning them down through longer acting G protein mediated pathways, okay? So here's the serotonin, and all I want you to notice here is that there's certain locations, and the happens to be a lot of them are in your brainstem, in the middle of your brainstem, right in the center of your brainstem, called the tegmentum of your brainstem in the center. There's these groups of nuclei. Don't have to know them now. You will eventually. But there's groups of nuclei that tends to be where a lot of cell bodies sit, and they project their axons throughout the brain. Some of them project them down into the spinal cord. But the cell bodies are in the brainstem, but they project axons into diffuse areas of the nervous system. And they basically squirt serotonin throughout lots of areas of the nervous system, or they squirt norepinephrine through lots of areas of the nervous system, or they squirt acetylcholine or dopamine to certain regions of the nervous system. And it's the balance of how much serotonin, norepinephrine, acetylcholine, dopamine you have in different regions of your nervous system at any given moment that is the background set point for your nervous system. Does that make sense? So when one of those gets off whack, something gets, that's when you have depression, or you have schizophrenia, or you have drug addiction. Those kind of things are all influenced, but when these get off whack, these are the things that are influencing larger networks of neurons because the cell bodies are down mainly in the brainstem, but they project their axons to influence larger areas of the nervous system, okay? Does that make sense? So now we're just going to go through and touch on some of the individual neurotransmitters and neuromodulators. Just talk about them as an individual a little more detail. So acetylcholine plays a major role in transmitting information in the peripheral nervous system, okay? Both at the neuromuscular junction and also in your autonomic nervous system, okay? In this central nervous system, though, it does play a role as a neuromodulator, okay? So think about acetylcholine as a neurotransmitter in the peripheral nervous system, especially at the neuromuscular junction. And when we get to autonomic nervous system, you'll be seeing acetylcholine coming into play here when we talk about, muscarinic, nicotinic receptors and all that stuff and how it influences responses of the target tissues that it's innervating, okay? GABA. GABA is a neurotransmitter, okay? Fast-acting central nervous system. So gamma aminobutyric acid, GABA. It's the major inhibitory neurotransmitter in the CNS. GABA is the major inhibitory. It's going to cause chloride channels to open and cause negative charges to go into your cells to make the cell less likely to depolarize. Okay, so it's an inhibitory. It's an amino acid. It will fall on either GABA A or GABA B receptors, are the two primary receptor types that it falls on. GABA A tend to be mostly located in the brain and brainstem, okay, GABA A receptors. And this is where it causes those chloride channels to open and more negative charges to come inside the cell. Drugs like Valium and other barbiturates and so forth that depress the nervous system tend to work on, they're GABA agonists. They act like a GABA. They act like GABA because they tend to bind to GABA receptors, and they tend to bind to GABA A receptors. The problem with when you bind to GABA A receptors is because they're in your central nervous system and they're in your brain specifically, is it also makes you sleepy and tired and can't think straight and so forth. So when your patients have a lot of I have patients who have a lot of tone or spasticity, and they're trying to decrease the activity of the nervous system. If you give them something like Valium, it makes their tone better, but also makes them so drowsy that they can't wake up to do therapy. So that can be a problem. GABA B, here's a little exception to the rule. Remember I said that most these neurotransmitters are fast acting mostly? Well, when GABA falls on GABA B receptors, which happen to be mainly in the spinal cord, they actually influence ion channels via second messenger system. It's a little longer acting, and it's through second messenger. It opens up some chloride channels and more in the spinal cord region. Okay, there's a specific drug called baclofen. Baclofen, you can either administer it orally, or they actually stick a tube into your spinal canal and squirt baclofen in and around the spinal cord, and it falls on those GABA B receptors. It also causes inhibition, but it doesn't affect your brain as much. So the nice thing about baclofen is you can give it to somebody, you downregulate, you inhibit the spinal cord so that maybe have less spasticity or tone, but they don't have quite the fatigue or the mental impairment that you would if you were binding to GABA A receptors. Okay, so that's why they built this molecule called baclofen that's specific to GABA B receptors, so it would cause less drowsiness and so forth than something that but it doesn't, it's not pure. It's still, some of it binds to GABA A receptors, so even when you give somebody baclofen, a side effect still is fatigue, cognitive issues and so forth. But it's not. It's definitely, you get a bigger out of proportion response of the muscle tone and spasticity being decreased versus impairment of cognition and so forth. Glycine is a neurotransmitter. It's inhibitory, inhibits postsynaptic membranes, primarily brainstem, spinal cord. So this is more in the brainstem and spinal cord than it is in the cerebrum and brain itself. Okay, so brainstem so glycine is another inhibitory neurotransmitter, but primarily in the brainstem and spinal cord. It's an amino acid. I think I mentioned in here too, aspartate. No, don't worry about aspartate yet. It's excitatory. We'll mention it after glutamate. So glutamate is the major excitatory neurotransmitter in the CNS. So GABA is the major inhibitory in the CNS. Glutamate is the major excitatory neurotransmitter. Fast acting, opens ligand-gated sodium channels. The specific receptor that this protein channel or protein receptor is called is the NMDA receptor, the N-methyl D-aspartate receptor. The NMDA receptor has also been implicated for neuroplasticity and this thing we call long-term potentiation, which we'll talk about we'll talk about neuroplasticity when we actually get to the clinical neuro next fall. In the fall, we'll have a whole session on neuroplasticity and recovery of function. That's why we skipped the chapter in this particular book. They have a chapter on that, but we do that when we get to actually treatment of patients and we talk about neuroplastic changes, response to treatment, and so forth. So remember that NMDA receptor when we get there. Interestingly enough, glutamate, if you get too much of it, you can overstimulate cells, make them work too hard and wear them out. So there's this concept they call exocytosis, not exocytosis, but excitotoxicity. Okay, so you excite it so much it becomes toxic. You create so much metabolic waste and metabolic activity that the cell can't clear it, and it actually just shuts down and dies. So, so what they're trying to do, so for example, after, let's say, a traumatic brain injury, especially like an acceleration, deceleration injury where you car accident, whack your head, traumatic brain injury, you'll shear a lot of neurons. And when those neurons shear, they just spill all their neurotransmitter that's currently in it out into the surrounding tissue. And a lot of neurons have glutamate in them. And so you just start spilling glutamate all around this injured area of vulnerable cells who already are like, crap, we don't want to work because we just got injured. And now you start spilling glutamate, and it makes them work too hard, and you can actually get this area around the initial injury, it starts to die off from this excitotoxicity. And so there's actually one of the reasons why they might do hypothermia on somebody and try to cool down their body temperature is to reduce the metabolic activity going on because of this excitotoxicity and extra glutamate. Or they might provide, steroids to the person or other medications that try to decrease the amount of glutamate damage and break down the glutamate so it doesn't cause this excitotoxic response. Okay. There was actually some fear that you could maybe, after somebody's had a big brain injury or stroke, you don't want to work them too hard because you don't want to make their neurons work too hard and squirt more glutamate in the area while they're vulnerable. However, most of that research was done with rats where they would do thousands of repetitions and force rats to do crazy things right after they lesion their brain. Most of our patients sit around for a few days and we're going in, we're not doing thousands of repetitions and making them work super hard. Most people have a big brain injury or big stroke. We're just doing basic sitting activities, positioning, range of motion. We're not making them do 10,000 repetitions of functional task practice, that kind of stuff that they were doing in these studies. So something to be aware of, but it's not something I think we need to be like overly concerned that we're doing the types of damage by making people work too hard, at least making the neurons work too hard, not talking like cardiovascularly and those kind of things, but okay. Talk about dopamine. So now we're talking about more of a neuromodulator with dopamine. Has effects on lots of different things. Dopamine influences a lot of nervous system activity. It does influence motor activity. And when we get to the basal ganglia, dopamine is squirted into the basal ganglia circuitry. And that keeps the basal ganglia working properly to set your overall kind of motor set point, how active your motor system will want to be at any given moment in time. So dopamine helps influence that through the basal ganglia. It also, though, plays a big role in motivation, cognition. It's part of your pleasure reward system. Okay, so dopamine. It's a monoamine. The major source of dopamine is the substantia nigra and the ventral tegmental area, the VTA. These are areas in the midbrain, in your brainstem. I have a picture here. If we go back to dopamine right here. This is your midbrain right here, and there's a region. The whole middle part of your brain stem, all down, like the meat of it, right down the center, like take a center cut, right down the, core. The core of your brainstem is called the tegmentum of the brainstem, okay? This area happens to be in the front part of that tegmentum, the ventral part of that tegmentum of the midbrain, called the ventral tegmental area, and that just happens to be where a bunch of nuclei of cells that have axons that project to different regions that squirt dopamine into those regions, okay? We're going to talk about dopamine as it relates to motor system, as it relates to Parkinson's disease when we get there. When we talk about the basal ganglia, we'll talk about what happens when you lose cells in that ventral tegmental area, and how does that influence the basal ganglia circuitry, and then how does that cause your motor system to have problems, okay? And we'll talk about those connections later. So, serotonin is another big neuromodulator. It's part of our big neuromodulating system here. Serotonin adjusts general arousal level for somebody. It also suppresses sensory information. So serotonin helps balance out and helps kind of filter out all the noise in the world, and also helps suppress some of the pain modulation and pain sensory. So people with chronic pain often have low serotonin levels. They're not able to filter out that chronic pain very well because they have low serotonin levels. We don't know if it was the pain that caused the low serotonin levels, or that they already had low serotonin levels and that makes them sort of susceptible to chronic pain, or now because of the pain they're not sleeping, which also affects serotonin levels. So there's lots of different factors that influence serotonin levels. But serotonin is important for giving you general sense of arousal and attention, but also suppressing extraneous sensory information, including pain. That's, not necessarily that we don't want all pain signals to come to us, and we want to be able to dull some of that. Serotonin helps with that. It also plays a role in the descending pain control system. So we have a descending pain control system. We actually send signals down to our spinal cord, so it helps to numb the impulses coming in initially to the spinal cord to modulate pain. Okay. We know that serotonin is definitely involved with depression, suicidal behaviors, and so forth. Low levels of that are correlated with depression and suicidal behavior. So it is also a monoamine. Neuromodulator, G-protein mediated, large networks of neurons that serotonin's influencing. Norepinephrine, also neuromodulator, provides a vital role, especially in active surveillance, active attention to our surroundings by increasing attention to sensory information. So it actually enhances sensory awareness and sensory attention. Serotonin balances norepinephrine in that respect a little bit. So you're always looking for, everything in life is a balance, this kind of optimal balance of these different neuromodulators based on the state that you're currently in and the current state that you should be in based on the situation. Highest levels of norepinephrine are associated with vigilance, lowest levels when you're sleeping. Your norepinephrine levels really drop down. They can bind to alpha and beta receptors. Norepinephrine is also very involved in the peripheral nervous system, in the sympathetic nervous system, the autonomic nervous system, the fight or flight response. So norepinephrine primarily acts as a neuromodulator, both in the central and peripheral nervous systems. But in the central nervous system, it has to do with more vigilance and attention. In the peripheral nervous system, it has to do more with autonomic, sympathetic function, okay? Fight or flight stuff. Also monoamine. Substance P. Substance P is a neuromodulator. This is primarily found in the spinal cord. Substance P acts as a neurotransmitter in the neurotransmitter in the spinal cord, as a neurotransmitter in the nociceptive pathway. At other sites, substance P acts as a neuromodulator. Okay, so in the spinal cord, more of as a neurotransmitter. In the other sites, in the central nervous system, as a neuromodulator, usually producing long-duration excitation of postsynaptic membranes. So it's an excitatory neurotransmitter that happens to be involved. We typically talk about it when we talk about nociception or pain pathways, okay, especially in the spinal cord. When the signals for pain first come into your spinal cord, go into the back part of the spinal cord, the first connection they make, usually substance P is involved with that first connection, okay, for transmitting that pain signal, and acts as more of a neurotransmitter in that. So when you bring in that first initial signal from the peripheral nerve and it goes into the spinal cord and it makes that first connection, which we'll get to next week, substance P is involved in that connection. It's a peptide, P peptide. We're going to group endorphins as a group of neurotransmitters. We're not going to go through, there's all different types of endorphins and so forth, but these are found in areas where we have opiate receptors, including the substantia gelatinosa, which is substantia gelatinosa is in the spinal cord, okay? It's actually in the area where we first receive pain signals in the spinal cord. So that's why if you have opiate receptors there, it tends to inhibit pain by binding to opiate receptors in the substantia gelatinosa and blocking pain signals when they come into the spinal cord. Also in the hypothalamus, the periventricular gray matter, which we don't need to know where that is now, but it's up around the hypothalamus area. And also the periaqueductal gray, which is a little bit of gray matter right in the cerebral aqueduct, which is running right down the midbrain into the pons. Remember that? There's a little bit of gray matter, which is cell bodies that have receptors for endorphins to bind to for pain. Mainly we're talking pain inhibition with the endorphin responses and opiate responses here, okay? Inhibitory. And these are G-protein mediated neuromodulators. They tend to have inhibition over the slow nociceptive pathways. When we get to sensation, we'll talk about fast pain. So when you whack your thumb or your finger, you get that initial shock like, boom, it hurts, move your hand, get away, and then you get the longer term throbbing pain, that's the slow pain pathways. The endorphins can kick in and try to down-regulate and modify and inhibit those slower pain pathways. This is just from your book here showing the neurotransmitter or modulator. So here we could call neurotransmitter or modulator. Their most common receptors, okay? So they definitely tend to fall on different types of receptors. As we talk about, move through the module or through the semester here, you'll notice like when we get to the basal ganglia and we start talking about dopamine and its role in the basal ganglia, we'll talk about what it means when it falls on the D1 receptor versus the D2 receptor. So you don't have to know that now, just know that there is more than one receptor that a lot of these fall on, and depending on the type of receptor they fall on can influence their action, okay? And some of these just receptors are located in different regions of the nervous system or in the neuraxis. Some can be more in the brain and the cerebrum, some can be more in the spinal cord, some can be more in the brain stem, some can be more out in the periphery. That can help define where your different types of receptors are based on their location. Let's do this. How many more slides? How many more do I have? Just two more? Okay, let's get through these two. Then we'll take a real quick break. And then we'll have one other thing. So just some clinical considerations. And you guys are doing pharmacology, right now? So you guys are going to be you're going to be applying this to your neuropharmacology. Because when you start talking about drugs that influence the nervous system, you're talking about drugs that are mimicking neurotransmitters is all they're doing, or they're blocking them, okay? So they're either trying to mimic a neurotransmitter and bind to the receptors that they're supposed to cause a certain action that's similar to what your nervous system would do, or they're trying to block those receptors or break down those neurotransmitters so they can't do that activity. So if they're trying to mimic it, they're an agonist, If they're trying to block or break down, they're antagonistic to that neurotransmitter, okay? So examples like botulinum toxin blocks the release of acetylcholine at the neuromuscular junction, so it weakens or paralyzes the muscles. So it's an antagonist to acetylcholine. So it breaks down or doesn't allow acetylcholine to be released to the neuromuscular junction. Only lasts for a couple months, and then the nerve recovers and is able to work again. But so if it's doing what you hope to do, you can just go back and you have to get an injection every few months. Baclofen, we mentioned already, is a drug that's given to bind to GABA B receptors. It's a GABA agonist, but it's specific to GABA B receptors, so it tends to inhibit the nervous system, especially in the spinal cord, and it's often given for spasticity or tone. Somebody has too much activity in their nerve or their muscles. They have excessive tone and hyperactivity in the muscle, they'll give them baclofen. Beta blockers, things for blood pressure, propranolol. It's a beta-1 antagonist. It blocks the beta-1 receptors that bind norepinephrine in the heart, okay? And so it blocks that, so it's a beta blocker, okay? This is what they call that. A drug called Sinemet we give for Parkinson's disease. It's converted to dopamine when it gets into the central nervous system, so it mimics dopamine. It's a dopamine replacement, essentially. So you give them this drug Sinemet made of L-DOPA. It's able to cross the blood-brain barrier as L-DOPA, and once it gets into the brain, then it's converted to dopamine. And it's a dopamine agonist, okay? Essentially it becomes dopamine and takes over that role for if somebody doesn't have enough dopamine. Things like collectively the selective serotonin reuptake inhibitors. So if you want the person to have, maybe they're having low levels of serotonin, you want more serotonin to stay within the synaptic cleft longer. So maybe somebody has low levels of serotonin, so you want whatever's in the synaptic cleft to stay in there longer so it'll bind to the receptors that are there. You give them a selective serotonin reuptake inhibitor so that you don't reuptake, it stays in the cleft longer. It doesn't get broken down as easily when it's been released. So you may have low levels of serotonin, but when you do release them, they stay in the synaptic cleft longer, so they're likely to do their job better and bind to the receptor sites more effectively. Does that make sense? So you're not adding serotonin into the system, you're just blocking the reuptake or the breakdown of it. You're slowing the breakdown of serotonin so it stays in the synaptic cleft longer, is what those serotonin reuptake inhibitors are doing. That's the same thing with MAOIs, monoamine oxidase inhibitors. So it blocks the enzyme monoamine oxidase from breaking down serotonin, dopamine, and norepinephrine. And actually, those are all monoamines, and so it's blocking the enzyme that breaks down all those monoamines in the synaptic cleft. And so MAOIs is a combination, is a drug that you'll see. There's different types of MAOIs, but when Dr. Renner gets there with you guys, that's all they're doing is they're blocking the breakdown of all three of those in the synaptic cleft. So, and that's why, you know, giving medications for depression, anxiety, and so forth there's as much of an art to a science to it, because everybody's chemistry is a little bit different, and everybody's response to those drugs is different, and so trying to find that optimal balance really is really a challenge initially, to try to figure out what that optimal balance of raising serotonin, lowering this, you know, and then anytime you change those medications, it often takes 1 to 2 weeks of any individual change of those medications, let alone multiple medications you're changing, to know how the long-term response is going to be. So, just a couple lists of some diseases that are common that you'll see that involve neurosynaptic transmission or neurotransmitters in some way. We mentioned Parkinson's and dopamine. There's a syndrome called Lambert-Eaton syndrome. It's a small cell carcinoma of the lung that actually, when they have this cancer, it releases antibodies to calcium channels that are distributed in the blood, and it impacts calcium channels so that you block the calcium channels so that you can't get release of synaptic vesicles. Okay, so it impairs nerve transmission via blocking of or damage to calcium channels. Myasthenia gravis, this is where you develop, it's kind of autoimmune, and you develop antibodies to the nicotinic acetylcholine receptors on muscle cells. So you damage the receptors for acetylcholine so your muscles don't fire as well. They get weakness in those muscles that are damaged. And then we mentioned depression, anxiety, all those kind of things, generally related to some abnormality or balance in levels of serotonin, dopamine, and norepinephrine.